So in this lab, we are going to try to understand this very interesting concept that is epiptasis. And here, remember, there is gene interaction. So one gene is affecting a second gene. In this case, one locus is masking or modifying the phenotype of a second locus. Okay, so in this case, we're going to use chromatography that we have used, that you already use in different labs, in cells energy or in diversity of life, as well as in chemistry. And in addition to that, we're going to use fruit flies as a tool, and we're going to use a lot of mutations. And you will see the mutations that we have studied, like the one that have white color, the one that look like wild type, and some of them that look like that have a different kind of reddish, and some of them that look like that have a brown eye. Okay, and what is the key thing that I want that you remember? So quickly, the main idea is that depending on, if you remember the beetle experiment, when he started using screens, here when you have a white eye, is that you are affecting that pathway in the beginning. Yeah, there will be true pigments that interact to produce the eye color. So if you have a normal eye color, you have reddish pigment and you will have a brownish pigment. Yeah, when you affect the pathway in the beginning, what you're going to get in the beginning is the white eye. So in the beginning, you will have this. Good. If you have a scarlet color, and I'm going to put this in the brownish. The scarlet is that you're affecting this part of the pathway, so this will be the scarlet. And if you have a brown eye, is that you cannot produce this part of the pathway. Okay, so this is key, and we're going to do that in the class. So how we're going to do it? Let me explain it very quickly how we're going to do it. So if you remember, we are testing this thing that you have studied in chemistry and biology. This idea, if you remember, of one gene of one enzyme, that I don't need to talk a lot about that, but remind you to study the things about beetle and tatum. The and in particular here, I told you that in a biological system, we wish that it was so simple like that. Actually, is more complicated, and instead of having one enzyme, you have an entire metabolic pathway when the substrate of one enzyme will end up to be, or the product of one enzyme will be the substrate of another one, and it will be affecting that. And for example, one example of that is the melanin. In our case, we're going to study something very similar, but using fruit flies. So to solve these problems, we are going to use Drosophila I, and in your lab manual, they talk about this. Why Drosophila I are so good for this, and the main idea is that they're very big and very easy to extract the pigment. So what you're going to do is, unfortunately, you have to kill these flies, and then you have to remove the head of the flies, and then you have to crush the eyes to extract the pigment. Okay, the in the video they describe how to do it, and in your lab manual describe to do it. There are some difference in the procedure that they ask you. So, for example, when you have the chromatography paper, to you cannot put it like directly into the solution. So I will show you in the video. They show you one way to do it. We do it in a slightly different way. Either way it works. The idea is that the paper doesn't fall in the liquid, and but it's much easier in the lab to explain how we do it. But the idea is that the videos are the previous video describe more or less all the steps of how to do this. Uh, Another piece of information that I wanted to tell you is that there are hundreds of mutations in Drosophila, and therefore the fruit fly eyes, there are so many, and we're going to use some of them to study this process. So like the white, the brown, the sepia, and there are other ones that we cannot study because there are too many, but the idea is the same. Yeah. So this is an schematic that show you a cell of a fly in the eye, and I want to remind you that there are many pathways. Yeah. Here you have enzymes, and the enzymes are coded by genes. Yeah. So for example, inside of the cell you have a pigment for the UV pigment, for the red pigment, for the yellow pigment, for the brown pigments. Yeah. And you have outside of the cell, sorry, uh, you have in the membranes of the cells proteins also that are not only in charge of the enzymatic exhibit, but transporting the substrates that need to go inside to produce all these pigments. Okay, so all these are genes. So what we are going to do is to have mutations affecting all these parts of the pathway, and in the based on that, for example, we are not going to have, if we have a mutation, we are not going to have brown colors, or we are not going to have red pigments depending on the mutation. So let me show you in a much simpler way. So here you have a simplified explanation of what is happening, and I want that you pay attention especially to this. Here you have the transporters, yeah, and all of them are working. So in the wild type, you're able to produce the red ones and the brown pigment, yeah, and this is why you have this red and brownish. And now I want that you see what happened in the next one. So here in B, you have this protein, this transporter is not working. So the brownish protein, the involving the produce the homochromes, the brown pigments are not working. So what are you going to get if the brown colors are not working? The brown pigment, you're going to get a, yes, a reddish eye, a scarlet eye. Yeah. So only the pigments that have the reddish colors are working. Now, here I want that you go to the component number six, number C, and here I wanted you to take what is the problem. The problem here is, yes, is that the reddish pigments are not working. So one is not working, you don't get the red pigment, so you get a brown eyes. Okay, because all these one are working, the ones that are in charge of the brown color. So now I have one question. What happened if the transporter number 2 is not working? What do you think that is going to happen? And in the lab, we will try to answer that question. Yeah, so in other words, we're going to try to identify it with chromatography and trying to identify in which component, in which you will find a defect. Yeah, so if you have, for example, a brown pigment, are you going to have the mutation here, or are you going to have the mutation here? Yeah, or are you going to have the mutation here? And you have to identify those things. So what we are going to do is to use chromatography, yeah, and take the fruit fly eye and have all this pigment here, and using the chromatography, we're going to separate the different pigments, and in that way we can see which pigments are present and which pigments are lost in each one of the mutants. Okay, so let me show you an example. So here you can observe an example of your results of tomorrow. So for example, here for the white eye, and let's start with a bluish here. What you observe here in number 1 and the wild type here, this is the pigment. Yeah, this UV pigment is here, but if you see the gene white is not there, and in the brown is not there, so it can tell you that maybe these were early in the metabolic pathway. And now let me take the red number 2 here, and you can see it again. It's in the wild type and it's in the scarlet one. But if you see, it's not present also here. So it can tell you more information of where it is supposed to be located. And you can do that with the different mutation and with the different information. Okay? And in addition to that, as I told you, you will observe this kind of. So as I said before to you, here this will be your results. Here you, for example, have females and males. You don't have to differentiate between males and females. And there will be so many pigments that they can be there. I add this schematic that show you the different pigments. We are not going to study all of them. We're going to study some of them. And here, this will be useful for you for your analysis. Okay, I hope that this was clear. Okay, I hope that this explanation was clear, that you have two pigments that produce the Drosophila eye color, and, and when you miss those pigments, the Drosophila eye can change, and we can observe that using chromatography paper. Okay, have a good day, and if you have any questions, just let me know. Bye.